Welcome to Lakshman Biology. Now we will discuss about the incomplete dominance. Incomplete dominance. What is the incomplete dominance? Before going to see about incomplete dominance, we have to see what is the law of dominance. In Mendel monohybrid cross, Mendel explained that there is a dominant character which will expressed in F1 generation. Okay, that is the law of dominance. According to Mendel law of dominance, what happened? Only one dominant character is expressed in the F1 generation. See here, Mendel crossed the homozygous dominant plant with homozygous recessive plant. When he crossed homozygous dominant plant with the homozygous recessive plant in F1 generation, all plants which are formed in F1 generation are dominant plants only, means which contain only dominant character, that is a tall. That is about the law of dominance. But if you see the incomplete dominance will not follow the law of dominance of Mendel. In incomplete dominance, there is two alleles are there. Two alleles are there. One allele is called dominant allele, and another allele is called recessive allele. Two alleles are there, but dominant allele cannot express its phenotypic character in F1 generation. Instead of expression of dominant character, the F1 generation plant will express the intermediate character which is present in between the dominant and recessive alleles. That is about the law of dominance. Example for law of dominance: flower color in Snapdragon. Example for it is a flower color in Snapdragon. Okay, if you see the flower color in Snapdragon, the dominant flower color in Snapdragon is red. Red is the dominant character. Genotype of red color flower plant is a capital R and small r. Then white is a recessive phenotypic character, and genotype of white color flower is a small r and small r. Red is dominant character, white is a recessive character. Then if you cross the red color flower plant with white color flower plant, we can see the formation of same type of gametes in both. Then we can see mixing of both gametes, which leads to formation of F1 generation. The F1 generation genotype is capital R, small r. This is F1 generation. But according to Mendel, what happened? The F1 generation plant should be express only one parental character, that is dominant character. But here, instead of expression of red color, but dominant allele is there, but dominant allele failed to express its phenotypic character in F1 generation. It will fail. Instead of red color, F1 generation flower will become into a pink color flower we can see in the F1 generation. When we cross the F1 generation plants that are heterozygous dominant plants, then we can get the F2 generation. Then if you see the F2 generation plants, the F2 generation plants has one red color flower, then two pink color flowers. Heterozygous condition will not leads to formation of heterozygous dominant, not leads to formation of red color. Instead of red color, which become into intermediate character, that is a pink color. Two pink color flowers are formed. Then smaller, this is white color. Here what happened? One red color flower, two pink color flowers, and one is a white color flower. Because of that, the phenotypic ratio of F2 generation of incomplete dominance become into 1:2:1. 1 is the red color, 2 is the pink color flowers, and the genotypic ratio also become into 1:2:1. If you see the Mendel monohybrid cross, the phenotypic ratio is 3:1 and the genotypic ratio is 1:2:1. But if you see in incomplete dominance, both phenotypic ratio and genotypic ratio is 1:2:1. In the genotypic ratio, 1 is capital R, 2 is capital R small r, 1 is small r. In phenotypic ratio, 1:2:1. 1 is red color flower, 2 is pink color flower, and one is a white color flower. Thank you. For more updates, please subscribe Lakshman Biology.